Okay, I'm chromosome number 1.
No, I'm chromosome number
fine.
1. I'm chromosome number 1.
And I'm chromosome number 2.
And we're here to talk about
Meiosis. There are 4 phases of interphase, the longest stage in mitosis. The stages are G1, S, G2, and M. G1 is the phase where the cell grows, preparing for cell division. The S phase is the period where DNA is synthesized. Chromosomes are replicated. The G2 phase is the phase prior to mitosis. The cell synthesizes the proteins, continuing to increase its mass. The M phase is the beginning of mitosis, where cell division starts.
Prophase is the stage of meiosis where chromatin condenses into chromosomes. Spindles, these, form once the nuclear envelope breaks down and forms poles. The centrioles move towards opposite poles due to the lengthening of the microtubules forming between them. The chromosomes then migrate towards the center of the cell.
Metaphase. The spindle, shown in yellow, fully develops and the chromosomes are in alignment. The nuclear membrane disappears and the chromosomes attach to the polar fibers from both sides of their centromeres, shown in orange.
This is the phase of metaphase.
This is metaphase. Go.
The paired chromosomes, the sister chromatids, separate and move toward opposite poles. Spindle fibers lengthen and elongate the cell. A complete set of chromosomes has assembled at each pole of the cell. This is the phase of anaphase. During telophase, polar fibers continue to lengthen. Nuclei begin forming at the opposite poles. Cytokinesis begins to occur as the cytoplasm divides, creating 2 distinct daughter cells.
Crossing over. Crossing over occurs in prophase 1. Crossing over of homologous chromosomes is what gives rise to genetic recombination.
The legs of the chromosomes cross over one another, creating a new variation. We are chromosomes. Right now in the nucleus, chromatin is condensing into us. During metaphase, the chromosomes are aligned at the center of the cell and spindle fibers attached to the centromeres of each chromosome, enabling them to be split. This is during anaphase. Right now, we are being attached to spindles at opposite ends of the poles. Okay. This is the last stage in mitosis called meiosis. Right now, the cytoplasm is dividing into 2 separate daughter cells. At the end of meiosis 2, 4 daughter cells are produced. Each of these daughter cells are haploid. Each of the 4 daughter cells have 1/2 the number of chromosomes of the original parent cell. Oh, I didn't see you there. I'm here to talk about disorders. Chromosomal errors happen as a result in error in cell division. Sometimes an egg or sperm ends up with too many chromosomes or not enough.
But what happens to the baby?
Well, the baby could have a trisomy or a monosomy, which is when the baby has an extra or a missing chromosome. An error can occur during a mutation, changing the organism's trait from what it normally would have been. Mutation causes a cell to produce an incorrect protein during protein synthesis. This causes a change in the organism's trait.
Yeah. These are 2 ways meiosis creates genetic variability. Crossing over is one of the ways meiosis can cause genetic variability. The other way is random alignment. Genes representing different pairs of traits are segregated into gametes. This is why you have some characteristics of your mom and some of your dad. The male gamete is called the sperm, while the female gamete is called the egg. Hi, I'm chromosome number 1, and this is gray tone. I have brown eyes for my dad, while my mom has blue eyes. This explains how I have different traits. Mitosis is fun!
We hope you're paying attention during the video. Till next time.
Chromosomes out!